Good afternoon, everyone. With great pleasure, I welcome you all to the third seminar of the Research Seminar series of DBLS. Today, we have amongst us Professor B. Ravindran, Emeritus Professor, Institute of Life Sciences, Bhubaneswar, to deliver a talk on evolution of TLRs in primates by malaria and sepsis. It's an honor to have you here, sir. I would now like to request Professor Rishi Shankar to welcome our honorable guest with a bouquet. Thank you, sir. I would now like to request Dr. Ajay Karakoti to kindly introduce the speaker. Sir.
Good afternoon, everyone. Hosting research seminar series is a really great thing because we get to meet so many great people who are doing really good scientific research across the globe. And today is no different. We have a very eminent speaker here today with us, Professor Ravindran Balachandran, who is currently an emeritus professor at the Institute of Life Sciences, Bhubaneswar. He was also the director of this institute from April 2006 to about 31st August 2016. And prior to joining and heading this institute, he also worked as a scientist at the Indian Council for Medical Research for more than 2 decades. So there is a long research career which is backed by Professor Ravindran, and we get to hear all the learnings today. A microbiologist by training, Professor Ravindran studied at JIPMER, Pondicherry, and then Delhi University, and later in the Liverpool School of Tropical Medicine, UK, and University of Connecticut Health Center, USA. His research interests include immunology of infectious diseases such as malaria, filariasis, and sepsis, regulation of inflammation, and macrophage biology in infectious diseases. He has also been an active member of a large global consortium of investigators from various universities across the globe, such as UK, USA, Germany, France, Netherlands, and so there is a nearly decade-long working on immunobiology of metazoan pathogens as well. He was also a visiting professor at the University of Edinburgh, University of Bonn, Pasteur Institutes during the last 10 years. And he has published about 90 scientific papers in international journals. One thing to take heart from here, and a lot of learning for people like us, is that over the last 3 decades, he has successfully supervised 18 PhD students and 16 MD students. That itself speaks a lot of volumes about today's speaker. So without any further delay, may I now request Professor Ravindran to please deliver today's lecture. Thank you.
Thanks, Ajay, for this generous introduction. I was just trying to figure out what the background of the audience is going to be so that I can pitch it accordingly. And the impression that I got from Ajay and Rishi is that it's largely students, master's level ISU. And then just now I got introduced to a person who is an engineer. So I'll try and hard in a way that it becomes intelligible, but there's a lot of data for those of you who are interested to look at hard data. But at the end of the talk, you'll certainly go with a message without worrying a great deal about the data. So it's a postprandial session, so there are always issues post-lunch session. So I'll start asking a question so that I wake up as many people as possible. How many of you have heard of TLR? Okay, so I don't see far too many hands, so that's where I will start. So toll-like receptors are a set of receptors that you see on phagocytes. I'm sure phagocyte is something that you can understand in a generic sense. These are immune cells that are capable of swallowing pathogens in a very simple term. So there is a way by which the immune system functions to recognize a pathogen. An adaptive immune system is a sophisticated one which can recognize any particular pathogen after the second or the third or the fourth time you get in touch with, or the immune system gets in touch with. But then there is a whole system, what are basically called the innate immune system. These are mechanisms by which, in a non-specific manner, your phagocytes are able to recognize pathogens. But then there's a certain level of specificity even in this. For example, gram-negative bacteria have a component called lipopolysaccharide or endotoxin that is present in all gram-negative bacteria in a generic sense as a class of bacteria that are recognized by certain receptors on the surface of phagocytes, what are called toll-like receptors. TLR4 is something that, so it doesn't matter whether the immune system is looking at an invading pathogen, whether it's a salmonella or shigella or E. coli, so long as it is a gram-negative bacteria, the immune system is able to recognize. There are other TLRs which can recognize by certain other patterns that is present. For example, in gram-positive bacteria, lipotecoic acid or peptidoglycan is present regardless of which particular gram-positive bacteria that you are talking about. So the TLR2 is something that recognizes in a very non-specific manner any bacteria, gram-positive bacteria, even mycobacteria, which have peptidoglycan. So in general, innate immune response is something which is non-specific. At the same time, there is a broad level of specificity that is given by toll-like receptors. There are a variety of other receptors. For example, TLR5 is a receptor that can recognize flagellin. So any bacteria which has a flagella, which is made of flagellin, one of the proteins present in flagella, will get recognized by phagocytes through TLR5. So this as a basic background of TLR, and very broadly all these receptors on the surface of phagocytes are classified. Some of them are not necessarily on the surface, you see them in the cytosol, are called pattern recognition receptors. Essentially, your immune cells are able to recognize quote-unquote patterns on the surface or inside the bacteria or any pathogen. There are viruses which can be recognized by the innate immune system. That is because all viruses are very rich in certain DNA sequences, what are called CpG motifs. And by that mechanism, the immune system is able to recognize an invading virus, and so on and so forth. So one of our interests is how exactly these pattern recognition receptors or the toll-like receptors are evolved. It's nothing unique to humans, it is nothing human to even mammals, to mice. It is something that you see in plants in a very rudimentary manner, and you see them, as a matter of fact, the toll-like receptors were discovered in Drosophila. It's a fly. So there is a receptor that was known to be in Drosophila immune cells, the toll receptors, and then it was recognized as something primordial to all the living creatures, starting from extremely low levels in biological evolution up to humans. And one of the things that we have been interested is to identify and recognize the importance of these receptors. So when you say receptor, this is this protein or a glycoprotein molecule which is sitting on the surface of the cells. Some of them are also present inside the cytosol, as I said a minute ago. And they do the function of getting activated or activating the cell when they come in contact with a ligand that is present in the invading pathogen. So in that sense, lipotecoic acid or peptidoglycan in gram-positive bacteria, lipopolysaccharide in gram-negative bacteria are the ligands. And for TLRs or the toll-like receptors on the surface of phagocytes are the receptors. So when this ligand-receptor interaction happens, a whole variety of intercellular cascade of activation happens, as a consequence of which the phagocytes gets killed. It means multiple things to multiple people. It gets activated, it gets angry, it can phagocytose better, it produces a whole variety of molecules that can eventually kill the invading pathogen. But there are differences in the way the toll-like receptors have evolved from a fly like Drosophila to humans across the species. And there is a second component to it. All the receptors in all the human beings are not identical. There is a very high level of polymorphism. The toll-like receptors are pretty common in all of us, but at the same time there are unique mutations that you see in them. And one of the reasons why we wanted to look at these mutations in the toll-like receptors was whether or not these mutations do make a difference in terms of their ability for functional difference, and whether or not these mutations happened as a consequence of pathogen during evolution. I don't want to say more about it at the moment, but as we go along, you will be able to figure out yourself. So this is just a cartoon showing how viruses, gram-positive bacteria, gram-negative bacteria, fungi, or protozoa. There's a whole long list of TLRs. You don't have to really read all of them. It's enough you take home the message that there are pattern recognition receptors that are able to recognize different pathogens in a certain specific manner, but not necessarily with a high level of specificity that you see in adaptive immune response. The second concept that I want you to recognize is innate immune response, as I was talking about, through TLRs. You don't have to worry about the cartoon. It simply shows this sequence of events that happens intracellularly when you have this ligand and receptor linking each other. And the consequence of this activation is pretty similar, pretty comparable to what exactly happens in the process of inflammation. Inflammation, I'm sure, is something very commonly understood by many people. It unfortunately means different things to different people. A physician or a clinician would have a certain concept of inflammation, which is completely different from what a cell molecular biologist thinks about and what an immunologist thinks about. But more than 100 years ago, pathologists described the process of inflammation. If you take a needle or a nail and then scratch yourself on the skin, within the next 5 minutes you get a wheal and flare formation and you get a red streak, and that's basically inflammation as a phenomenon. But at the molecular and the cellular aspect, what happens is this. You can either have molecules which are coming from pathogens, you can also have molecules that are endogenously coming from your own system, what are called danger-associated molecular patterns, which also use pretty much the same set of pattern recognition receptors, the toll-like receptors, and activate the cells, and which they produce a whole variety of molecules what are broadly classified as chemokines. They attract cells, and they also indulge in dilatation of the vessels, as a result of which you get the redness, what you see as inflammation. But essentially, at the molecular and the cellular level, innate immune response is no different from an inflammatory response. So these are two sides of the same coin. It's enough that I talk about this at this stage, that the toll-like receptors mediated innate activation or innate immunity is the molecular cellular basis of inflammation. So there is a whole variety of TLRs that I talked about, but of all the toll-like receptors, there is one which is extraordinarily promiscuous, is a TLR2. TLR2, as I said, looks at gram-positive bacteria through their lipoteichoic acid or peptidoglycan, but they also end up seeing a whole variety of other molecules present in several other pathogens. They are listed on the left, starting with gram-positive bacteria, mycobacteria, and trypanosomes, parasites, Treponema pallidum, one of the spirochete which causes syphilis, and so on and so forth. So, and also more recent times, we have VI polysaccharide, which is present in Salmonella typhi, the bacteria that causes typhoid in human beings, is also a TLR2 ligand. And like GPI anchored proteins, glycosphosphatidylinositol anchored proteins that you present in plasmodia, that you see in plasmodia, are also capable of activating cells through TLR. So the only reason I have projected this slide is for you to appreciate that it is not so highly specific. There is a very broad specificity. At the same time, there is a large number of molecules that can be recognized by these pattern recognition receptors. But in that context, TLR2 would probably beat all other TLRs because they are far more specific. TLR4, for example, recognizes lipopolysaccharide present in gram-negative bacteria, and then a few other molecules present in other pathogens. But TLR2 binds to a whole variety of them. You don't have to read this slide completely, just read the title. It's enough. So what it says is there are polymorphism in the gene of TLR2 in humans. In other words, when you talk about polymorphism for an engineer, if I have to say that everything is identical between the TLR2 of all of us here, but then there are some sequences, some residues which are mutated in a manner that is different from each other. Fundamentally, they process very similar in all of us, but then there are qualitative and quantitative differences by which my TLR2 would recognize as opposed to somebody else's TLR2, and that is mediated by small levels of polymorphism or mutation. Now, these mutations don't come all of a sudden. These mutations come through evolution, and one of the things that I'm going to talk about is a short story of the lab that has emerged in the last 3-4 years in identifying certain mutations, and a mutation that is of critical importance to the functioning of TLR2, and that's what I'm going to talk about. I'll pass this. I'll now come down to narrow down from all of TLRs to TLR2, and in TLR2 there are a variety of polymorphisms, and one of the polymorphisms we recognized, which was identified by us about 4 years ago, is a 23 base pair sequence that the whole chunk of this is deleted from one of the exons in TLR2. This whole sequence of 23 base pair is deleted in some of us. What percentage of us, I'll come in a minute. But by and large, insertion is seen in a vast majority of human beings, approximately about 70 to 75% of us. So if I were to type the TLR2 polymorphism, this sequence of deletion of 23 base pair, in all of us in the auditorium, about 30% of us, approximately, there is a reason why I keep emphasizing approximately is that I'll come down to more specifics as I go along, will have a certain deletion mutation that will be absent in your TLR2 gene as compared to the 70% of the individuals who will have the insertion. So one of the reasons why we have been busy with is to understand what this deletion actually means when it comes to the functioning of TLR2 in these two groups of people. This is exactly what I talked about. Regardless of which geographical area, which ethnic population you look at, we have looked at it in Bhubaneswar cohort, we have looked at it in people in Delhi, and then there are others who have recently looked at in Papua New Guinea, in Kenya, North America, where people of African origin or Caucasian origin or Asian origin, you see pretty similar, but there are mild differences in the percentage of people. But vast majority of people have the insertion. That's why we call it the wild type, and the mutation or the polymorphism is seen, the deletion mutation is seen in a smaller percentage of people. This smaller percentage can be as low as 20% in some ethnic population, it can be as high as 35% in other ethnic population. But you see them in all human population. there is an evolutionary sequence that is related to this mutation. The top two rows is what you see as humans, where the insertion mutation is seen. The sequences that I have shown in yellow is the insertion, which is what 70% of the people in the hall may have. And the dotted line is where the deletion is, where this chunk of DNA is absent in 25-30% of the people. But go down the species, in gorilla, in chimpanzees and orangutan, these are the higher species, primates and more evolved primates, you see that all of them have, I should qualify, not all of them. This sequence is taken out from the genome database. As you know, the genome database comes from a single animal. The DNA has two strands, and essentially the genome database, you only sequence one strand, and that is what is uploaded in the genome database. So what you see here is that all those who have, the insertion is present in the gorilla sample that was typed by the genome bank and then loaded. It has the insertion, not just one, like the way 70% of humans have. It has two such insertions of 23. And chimp has identical to the vast majority of humans, 30% as I said, who have an insertion. And the orangutan also has the insertion. But if you go down in marmoset monkeys and namascus, rhesus and baboon, the lower primates which have been around for far too many years, the deletion is seen. So in other words, let's not take this as a gospel. This is a sequence that you see if you search in the NCBI database for the insertion deletion. This is what you will see. There is a certain hierarchy of lower primates having a deletion and higher primates having an insertion. And if you do it in humans, vast majority, as 70% as I said, will have an insertion. And it is actually true of even the Neanderthal. There are 3 bones of Neanderthal. Neanderthal genome has been sequenced more recently, and at least there are 3 sequences which are available, and also shows very similar to that of the higher primates, gorilla and chimp, which has the insertion. So what does this sequence difference in the TLR2 mean when it comes to activation of cells? So if I take my cells, I have an insertion and somebody else has a deletion, and then I score for TLR2 on the surface of monocytes of the 2 individuals or 2 groups of individuals, and those who have an insertion will have less number of TLR4 expressed on the surface, which you can actually score by flow cytometry precisely. And those who have deletion tend to show a higher expression of TLR2. So this flow cytometry histogram that you see in pink is the one from deletion individuals, and the one in green is from the insertion individuals, and blue, of course, is the background negative control. So if you do this, the left histogram is done for a given individual, a representative one, and the right bottom, what you see is the expression of TLR2 in individuals who are insertion, which is red, and all the blue dots are, every dot stands for one individual, where you see a higher expression of TLR2. It is not only TLR2, but there are 2 other molecules that are present in plasma. This is something which many of you would know, the C-reactive protein and also TNF-alpha. Both of them are inflammatory molecules which you can measure in circulation, in plasma, and which is an indicator or a window of demonstrating that somebody is having an inflammation. So it's a systemic inflammation indicator. If somebody is having rheumatoid arthritis, chronic inflammation of any kind, systemic lupus erythematosus, and any of the autoimmune diseases, you will see the clinician would ask for a C-reactive protein or a TNF if you can afford. It is not routinely clinically done. These will be high in individuals who have a systemic inflammation. And the point I'm trying to make here is the blue bars that you see here and here are the levels of TNF-alpha and C-reactive protein if you happen to have a deletion mutation. The red bars are those who have an insertion mutation. And it also demonstrates that the level of the TNF and C-reactive protein in circulation is directly related to the level of TLR2 that you express on the surface, as shown by flow cytometry. And that's exactly what the lower panels demonstrate. If you score for C-reactive protein versus TNF-alpha and surface TLR versus TNF-alpha, all of these give you a positive association, suggesting that if you have this mutation of deletion, 23 base pair, you end up expressing more of TLR2 and you end up being relatively in a higher inflammation state. You don't have to worry about this. All this tells you is that regardless of this mutation, your other receptors are comparable. Now, you can take the cells of these two. What I had shown you data so far is that individuals who are having a deletion will have a higher level of C-reactive protein or TNF-alpha. But what happens if you take the cells of these individuals with deletion and with the insertion and then kick them in vitro, stimulate them in vitro with a TLR2 ligand? So this is what happens. There are two different TLR2 ligands that are used here. One is PAM3CSK, which is not a natural ligand, naturally existing, but it is a synthetic, artificially made molecule that mimics a TLR2 ligand, like a peptidoglycan. And it also shows VI polysaccharide, which I said is present in Salmonella typhimurium, which is a TLR2 ligand. So compare the blue bars and the red bars. The blue bars are those with a deletion mutation, the red bars are those with an insertion mutation. Very consistently, and then you are scoring for production of TNF-alpha, IL-10, IL-1 beta, IL-6. You don't really have to see all four of them. If you see one, you have seen all four, because the trend for all the molecules are very comparable. If you have a deletion mutation in your gene for the TLR2, you end up making more of IL-6 and more of IL-1 beta, more of TNF-alpha when you come across, or when your cells come across, a TLR2 ligand. And that's actually not true for a non-TLR2 ligand. And that's where the CpG motif, well, CpG DNA is a ligand for TLR9. So regardless of the deletion or the insertion of TLR2 gene, you still end up responding very similar to a TLR9 ligand, which is CpG motif. What I'm going to show is a set of data identical, pretty similar to what I have shown you here. So, except that it is done in 3 different classes of people. Those who are homozygous for insertion, the orange bars, the tiny ones, and those who have a deletion which is a heterozygous mutation. In other words, in one of your DNA strand you have an insertion, in the other strand you have a deletion, and that's a heterozygous. And in both the strands, if you have a deletion, that is what shows here as a deletion homozygous. So when you have said that 70% of the people have insertion, 30% of the people have deletion, if you look at the percentage of people who have deletion, it will be extremely small, because the chance of a heterozygote itself is 30%, and a homozygous insertion, wild type, is 70%. The progeny likely to have deletion becomes extremely less. So you get something like 1 to 2% of the population will be having this kind of a deletion mutation. So what you see here is there is a hierarchy of activation. If you have a double deletion, you end up activating your cells much more compared to a single deletion as compared to a wild type. You don't have to worry about it. I'm going to skip this because this is pretty much true for a whole variety of ligands that you can take on. So what I have shown you so far is that humans with a deletion allele in TLR2 display more surface TLR2 and higher levels of inflammation in plasma, as shown by C-reactive protein as well as TNF-alpha. And their monocytes get activated more vigorously when stimulated with TLR2 ligand. I've also shown you data from the database that lower primates display deletion, at least in these cases. The database is a representative one animal. I'll come to that more detail in about 5 minutes from now, while the higher primates have an insertion allele. So what was the selection pressure during primate evolution that was responsible for introducing this insertion allele? So imagine when you say that rhesus monkeys have a deletion, something must have happened. Evolution selection always happens as a consequence of an evolutionary pressure, except that in evolution, somewhat like what the astronomers or astrophysics would say, everything runs into millions and millions of light years, or in this case, in several million years. So for example, the lower primates, from that the higher primates evolved. The old world primates evolved sometime around 25 million years ago. Chimps and gorilla evolved about 10 to 15 million years ago. And humans ourselves, as a human species, we evolved approximately 2.6 to 2.8 million years ago. So something happened during evolution from the lower primates to the higher primates, that there was a requirement for putting this insertion of 23 base pair in these animals. And one of the questions we wanted to find out was, what could have been the selection pressure that would have forced the selection or the insertion to happen, as a result of which the inflammation was lower in the higher primates. And malaria is one disease which has been constantly proposed for selection in evolution. For example, there are several hemoglobinopathies. How many of you have heard of the word hemoglobinopathies? Okay, Vivek knows it. So hemoglobinopathies are genetic disorders in hemoglobin as a consequence which has evolved during evolution of humans. For example, there are beta thalassemia patients, there are alpha thalassemia, glucose 6-phosphate dehydrogenase deficiency, sickle cell anemia. All these are defects in hemoglobin or red cell morphology in one way or the other. How did they come around? They came around because this is what the J.V.S. Haldane proposed 50 years ago, but what he said was very simple. In areas where malaria was highly prevalent, as it happened in Africa, and in several other countries in the tropics where the malaria has been prevalent, the selection pressure was, there was an advantage, say for example, sickle cell anemia. Just like the mutation that I was talking about, heterozygous and homozygous and the mutation. So if you think of an individual or a group of people with, say, beta thalassemia. So you can have individuals who are homozygous for thalassemia, who are normal, and then there is a percentage of people who are heterozygous. It is true for sickle cell anemia. You can have a sickle hemoglobin. Sickle hemoglobin, for example, happened as a consequence of a single mutation in the globin gene. So as a result of which sickle cell anemia happens in these people. If you have both the strands of your DNA having the mutation, then what you end up having is a kid born as a homozygous for sickle cell hemoglobin. The chances of survival of somebody with a homozygous sickle cell hemoglobin is extremely low, as a result of which, before even attaining the reproductive age, the kid dies. But on the other hand, those who are heterozygous, where the mutation is present in one strand and not in the other strand, you get a sickle cell hemoglobin, which gives resistance not specifically to malaria. In other words, a kid having a homozygous sickle cell hemoglobin will die because of the severity of hemoglobin nature, but somebody with a heterozygous sickle cell hemoglobin can survive And they survive actually better because it gives protection against malaria. So if you imagine a community of 10,000 people where there's a small percentage of people with sickle cell disease, kids with homozygous disease, they would die. And there is a percentage of people who are heterozygous for sickle cell hemoglobin, but who are protected against malaria. Imagine about 100 generations of this population of 10,000. All those who had the heterozygous mutation would survive and live longer because of, without having to suffer malaria, because the wild type who have completely normal for sickle cell and hemoglobin will be susceptible for malaria and they would die. In evolution, you have to remember there is no such thing as treatment. The treatment for diseases is 200-300 years old, at best 500 years old. So for 2.8 million years, we have evolved without treatment. If you get malaria, if you survive, you survive. If you don't survive, you die. So it's as simple as that. So death due to malaria would select out population which will have a survival advantage. And in the case of hemoglobin, sickle cell hemoglobin, this is exactly what happens. People with homozygous wild type will get disease. People with homozygous sickle cell hemoglobin would anyway die because of a deficiency of hemoglobin. And those with heterozygous are the ones who will survive better malaria. As a consequence, they are the ones who will reproduce more. And over several thousand years, the individuals with hemoglobin gene, sickle cell hemoglobin deficiency gene, will get enriched, selection by malaria. So this is the crux of the talk that I'm going to be giving you in the remaining 20 minutes. A mutation happens by accident or otherwise. All mutations happen by accident, by chance. In evolution, as a rule, all mutations are not by design. It is always by accident, by chance. And then whether that accident helps the species to survive longer, whether that accident helps the species to reproduce long enough by survival, would determine whether it is a beneficial mutation or otherwise. So this is how natural selection works, and almost all mutations happen by accident, but they get selected out if it was advantageous, or otherwise the mutation goes off along with the species. So we ask the question whether or not malaria would have been responsible for this insertion to happen, because those with the deletion, our working hypothesis was those with deletion have a survival disadvantage because they will be inflamed more and their chance of survival against malaria could be less. So this is exactly what we have data to demonstrate possibly malaria selected out. I'll take you very slowly through this. The left top is what you see in different clinical categories of malaria. I'll read it out for you. If you have seen the left part of the panel, you don't have to see the right part, because the one on the left is TNF, the one on the right is C-reactive protein. And very broadly, the picture is pretty comparable. So there are 5 different groups of people. Cerebral malaria is the first one here. Multi-organ dysfunction. This is the worst form of malaria one can think of, where people die of multiple organ failure. The third one, NCSM, is the non-cerebral but severe form of malaria. The fourth is UCM, or the non-complicated malaria. Malaria, normal ones that you see people in an OPD, and the HC, or the healthy controls. And you are scoring for TNF in plasma. And you need to remember all of these are drawn in log scale. This is 10, 100, and 1,000. So the difference is very, very significant. And regardless of what it demonstrates, you look at only the left top. If the severity of the disease is very high, the plasma TNF is very high. And if the severity is very high, that's where the first, second, third groups have very high levels of these molecules as compared to the fourth, and the fifth is a control. And there is also an association between death and survival depending on the levels. So the bottom left, if you see, the red dots are those who did not survive, who died. They all had very high levels of TNF, and the blue dots are those who survived malaria, regardless of the clinical severity, but it's just survivor versus death. So those who had very high levels of TNF had bad prognosis and they died, and those who were blue dots are those who had malaria but who survived and they had lower TNF. Now you ask the question, you reshuffle this, ask the question, how many of them have this deletion and how many of them have this insertion between the different groups? So what you see here is this. The blue dots are those who have deletion for homozygous. They had both deletion, both the strands deletion mutation, and have the highest TNF. In other words, if you have the deletion mutation and if you happen to get malaria, the chances of you making very high levels of TNF and the chances of one's survival is compromised. So what it shows is that the deletion mutation was knocking out people by malaria during evolution. Even today, although you have people with deletion and insertion, although we have chemotherapy, we have treatment, many things come into the picture. That's why once you have treatment, you have interfered with the natural selection. The evolutionary pressure selection doesn't happen once you have treatment. So, but then we have evolved for so many years without any such intervention. It's only in the last couple of 100 years we are able to block evolution in one way or the other by treatment for different diseases. Otherwise, the natural course would have been those with deletion, many of them would have died. What it shows is, even with treatment, if you have a deletion mutation, the chances of survival actually is low if a person gets malaria. This is pretty similar to what, even if you break it down to different categories, in each of the categories, the blue bars are the tallest, essentially telling you that if you have a deletion mutation, in every clinical group, the mortality is very high. It's a busy slide, you don't really have to read all of it, you just take a look at the ones that are highlighted in red. What it tells you is that if you have a deletion mutation, homozygous deletion, the chances of you getting multi-organ dysfunction is 13.5 times more than a person who's having insertion. In other words, the odds ratio essentially tells you what are the chances of somebody getting a certain disease or certain consequence as a result of a mutation. And what it tells you is that once you have a deletion mutation, the chances of multi-organ dysfunction is 13 times more than somebody with an insertion. So all that essentially giving you an idea, this is exactly what it strengthens the fact that survival, if you look at the odds ratio, the deletion has 4 times more chances of dying due to malaria if they have a deletion homozygous mutation as compared to a wild type. The next disease that natural selection actually knocked out people was due to sepsis. I don't want to go into the depth of everything about sepsis. It's already 40 minutes into my talk. I'll take probably 3 to 5 minutes before I wrap it up. We can take questions. Essentially, sepsis is again a disease where if you look at severe sepsis, the chances of the deletion individuals surviving sepsis is actually high as compared to those who are wild type. That can be checked even with the TNF levels, plasma inflammatory cytokine levels, TNF, IL-1 beta, IL-6, where the deletion individuals with sepsis will be far worse, the red bars that you see here as compared to the blue bars. So what I have shown data so far is that severe malaria and sepsis are associated with high inflammatory response, higher mortality, as well as higher frequency of patients with deletion allele. The database reveals, as I told you some time back, the database comes from a single animal. So how dependable is the database? Can it actually be verified in a large number of animals? And is it possible to do? It's never easy to do, but this is the repeat of the slide that I had shown you earlier. The yellow ones in the middle are the insertion that you see in chimps and orangutan, but this is in the database, as I said, in the single animals. And the dotted lines that you see below are marmoset and Nomascus rhesus monkeys, where there is a deletion. This is in single animals in the database. But we actually checked it in a large number of animals. We checked in 4 different rhesus. All of them had deletion. So in other words, the one that is in database in a single animal is actually true, at least in 4 animals. And 3 different baboons we tested, all of them were deletion, pretty similar to what the database showed. But we did something else. We managed to get fecal DNA from wild chimpanzees in West Africa from one of the collaborators, and there are first 5 animals that I'm showing you as Nandankanan C1, 2, 3, 4, and 5 are 5 chimps that are available in the Nandankanan zoo. But essentially, the 5 animals are actually one father, mother, and then 3 of the F1 generation. So it amounts to just 2 plus 1, 3 animals. But still, you consistently see in all 5 of them the insertion pretty similar to the database. But more important is the next 20 animals, chimps, different numbers, the GT series that you see, are all animals in West Africa where the chimp DNA was, basically it was a fecal DNA. You can't collect blood from wild chimps, but you can verify DNA of the animal from the stool. And that's where the samples came from. And it tells you that at least in 25 out of 25 chimps that we have tested, the insertion happens consistently, what was seen in the database. Similarly, we also checked the gorilla DNA of 8 different animals in the wild, and what it shows is the insertion that has happened in the database is actually true. So what I have told you so far is an evolutionary story that these 3 species, baboon or rhesus or Nomascus monkeys, have a deletion mutation, and the chimps or orangutan or gorilla or Neanderthal have an insertion mutation. But in humans, for example, I'm an insertion and Prakash, who works in my lab, is a deletion. And how is it possible in evolution that a species, lower primates had deletion, higher primates had insertion, but furthermore evolved than all of this, humans, we still have 70% or 75% of the individuals with deletion and with insertion, and the remaining 20-25% having deletion. So where did this deletion come into humans when our ancestors, although the numbers are small, 25 chimps or 8 gorilla or 3 Neanderthal that have been typed so far have an insertion, but we still have a very high percentage of deletion, very similar to the old primates. It can be due to multiple reasons. One of them is whether or not we tested sufficient number of primates, because 25 chimps in the wild is not necessarily normal, because they're all wild animals, grown-up animals. Probably if there were deletion, they would have died. So there was a certain selection bias when you are collecting samples from chimps in the wild. You're essentially picking up animals which have managed to survive, and those with deletion probably would have died, and you have missed. And it gives an impression probably that all of them are insertion, not necessarily true. And the second possibility is that about malaria. It is known that higher primates, when I say higher primates, chimps and gorilla, for example, more than 50% of them are infected with malaria with no disease. This is something which was reported 3 years ago in Africa. This it's already 5 years, 6 years. Nature paper published about the origin of human malaria. This has been repeatedly reconfirmed in the wild chimps and wild gorilla, that more than 50-60% of the wild animals have malaria, unlike humans. So, in other words, all of them had insertion. if you remember, that is why. So the assumption here, the evolutionary assumption is that insertion mutation happened. They were all deletion to begin with, and the insertion happened as a consequence of which they were protected against malaria. As it happens in the wild today, more than 50% of the chimps and gorilla are naturally infected, otherwise completely healthy animals with no disease whatsoever. And that's not something that happens in humans because of chemotherapy. So what I will leave you with this, the humans with the deletion allele. So the question is still open. We do not know where exactly we acquired the deletion mutation in a small percentage of people. It could have been because of the fact that Plasmodium falciparum, the one that actually is lethal in humans, evolved more recently. And there are evidence to suggest that until about 6,000–7,000 years ago, 2.8 million years humans have been around as a species, but until about 6,000–7,000 years ago, there was no such thing as agriculture. So agriculture started 6,000–7,000 years ago in Mesopotamia, and Anopheles gambi in Africa, which is a vector for Plasmodium falciparum, is a rice field vector. So one of the things that is being proposed is that as a consequence of agriculture, as a consequence of rice cultivation, we ended up cultivating Plasmodium Anopheles gambi, one of the vectors of Plasmodium falciparum, for transmitting the disease, and as a consequence of which, only for about 6,000 years, probably Plasmodium falciparum has been in humans, and out of 2.6–2.8 million years, 6,000 years in an extremely small window. So essentially, humans lived without severe malaria for more than 2.6 million years, and Plasmodium falciparum, the killer disease, is a more recent phenomenon in evolutionary terms, as a consequence of which we possibly lost the insertion during evolution. All chimps and all gorilla acquired insertion, but humans lost out the insertion because of absence of malaria, because the whole argument is that malaria is responsible as an evolutionary pressure to bring in the insertion allele, and in the absence of which many of our ancestors lost, and we end up having percentage of deletion allele. So I'll leave you with this. All of this work was done by one of my graduate students who is now an assistant professor in Central University in Ranchi, Aditya Panda, and of course several of my colleagues and collaborators. Beatrice Hahn is the one at the UPenn who has been very kind enough to give us extraordinarily precious DNA from African chimps. And Tushar in CCMB is somebody who's a genomics person with whom I've been collaborating for quite a while. And Sudhir Majumdar is the primatologist in National Institute of Immunology with whom I've been having collaboration. And Krishna Prasad in National Institute of Medical Sciences is somebody who works on tuberculosis, and much of the work that I initially showed, I didn't elaborate heavily on that, on tuberculosis and TLR2 polymorphism, was done in collaboration with Krishna Prasad. Thank you. I'll be more than happy to take questions.